So good afternoon, everybody. Thank you, everyone, very much for attending today's IMAP monthly webinar on post-season data analyses and review. So my name is Douglas Knopf. I am the Invasive Species Project Specialist for the New York Natural Heritage Program on the Invasive Species Database team. I'm also joined today by Fate Sewan-Yuan, one of the data analysts on the NYNHP Invasives team. Just a few housekeeping things before we get started. Today's webinar is going to be recorded. After the webinar has finished, I'll be sending out an email to all registrants that has a link to the recording after it's been uploaded to our YouTube channel, along with a few links for anything that I mentioned during the presentation today. Everybody's microphone is muted, and I ask that everyone please keep their microphone muted through the duration of the presentation. If anybody has any questions come up during the presentation, you can go ahead and put those directly into the chat. Either Fate will take a look at them and answer them as we're going along, or we'll have time at the end to answer all the questions, or if people want to unmute or ask questions at that point, you can do so. And with that, we'll go ahead and jump into it. So for today's agenda, we'll do a little bit of introduction about NYNHP and IMAP invasives. We'll go into uploading data from your mobile data collection tools and a little bit about data quality assurance and quality control. Going through the process of confirmation of data, and then finishing off with the analysis of data and presenting the data that you've collected. So starting off with the New York Natural Heritage Program, what we are as an organization is the record keepers and data collectors for all of the rare, threatened, and endangered species and ecosystem type information throughout New York State. And as one of the greatest threats to rare species is the establishment and spread of invasive species, the invasive species database team was established under NYNHP, and we are the ones who facilitate the official invasive species database for New York State. We do that through the IMAP invasives database software, where we host the actual physical database itself, as well as a host of data collection tools, things like the Watercraft Inspection Stewardship Program application, mobile apps, Server 123 forms, etc., as well as a host of data analyses and prioritization models to help give partners useful views and analyses of the data that they are providing to us in order for them to better prioritize their invasive species surveys and management throughout the state. So starting off with the upload of data, so everyone has been out all throughout the field season collecting data with our various field data collection tools. And the first step to getting all that data to us is to make sure that it is not on device still. So most of our mobile and field data collection tools are designed to be used offline, so setting them up initially, being able to go out into remote areas without cell service or Wi-Fi connection to collect your data, and having it be stored on the device that you're collecting on, and then uploading it at a later date when you're back in the office and you have time to go through all the survey that you've been conducting. So step one to make sure that we get all the data is to go through all of your tools, all of your devices that you've been using throughout the summer, and making sure that none of the data is still stored on it. So starting out with the mobile app, very simple. If you see any data on the screen when you open up your mobile app, it means that it has not reached us yet. So the best way to go through and do that is just to select every record that you have on your mobile app, either by manually checking the box on each record or going into the menu and hitting select all, and then going back into the menu and uploading all selected records. It'll ask you if you're sure, and then everything will be gone. At the end of the season, what you want to see is just an empty green box when you open up your mobile app. That means all of the data has left your device and has made it to IMAP Invasives database. For our various Survey 123 applications, so that's our IMAP Invasives General Survey 123 form, our Rapid and Classic Forest Pest Data Collection tools, as well as the Simple Aquatic Survey Pro, SAS Pro tool. You want to go through and make sure that all of these surveys that you've been collecting during the season have been completed and sent. So on the screen here, it has an example of all of the different boxes that you might see. The initial data collection box for starting new surveys, Drafts is for any surveys that you have started but have not yet completed or filled out all the required information. So if you have things in your drafts, making sure that you go through, fill out everything, and save them properly. I have one draft here in my example. If you go into your drafts and you have one in there that has a little star next to it, that means it's for your favorite answers so that you don't have to go back in and fill in your name, email, user ID, and everything. So if there's just one form in your drafts that has a star next to it, you can leave that one there. Anything else needs to be completed and saved. If you have anything in your outbox, it means it also hasn't been sent into us yet, so making sure you go into those and send them in as well. And then at the end of the everything should be in the sent folder, so you can see all that's collected and sent to us successfully. If you have just sent it very recently, or you hit send and then go right on to IMAP Invasive to look at it, you won't be able to see it right away. All of the data that comes in from server 123 it goes through a data repository on ArcGIS Online first. It gets put into a data layer there and then is crosswalked over into the database proper on an hourly basis. So if you sent anything that was still remaining in your server 123 app, give it an hour or so before you go into the main database to check and make sure it went in properly. And then for our final data collection tool, we have IMAP Mobile Advanced, IMA. This is the field maps based tool that is one of our map-based tools, so it's commonly used by professionals in the field. This tool is a little bit unique in that it is hosted on a live data layer in ArcGIS Online, but you can have offline areas if you're going to be working in those remote areas. You can also have multiple people on multiple devices uploading data into the same searched areas or on the same sites. So the most important thing is to get all of the devices that have been used in the field. So if you had a field crew and multiple people using multiple tablets or smartphones, getting all the devices together and looking through all the offline areas and making sure that they are all synced to the main online area. Best way to do this is to have your whatever mobile device you have in front of you, and then having an instance of AGOL's IMA layer running computer live and connected to the internet, and making sure that all of the data that is visible on mobile devices is also visible on that live layer. If anything is missing from the live layer, but you can see it on your device, it means it hasn't synced properly, and you just want to sync up those offline areas. Once you have everything off of your mobile field data collection devices, you'll want to go through and to make sure that it will be ready for crosswalk into the main IMAP Invasives database. The way to do that is to mark it as ready for upload. You do this by going into the searched areas at the main purple box for whatever site you were doing surveys management at, and at the very bottom of all the different data fields, there's a box that is labeled ready for upload into IMAP 3. Once you've gone through and reviewed all of your data, which we'll talk a little bit more about in a second here, gone through and you do your review, and after that is done and you are sure that you are finished with collecting data at this site for the season, you'll change that ready for upload box to yes, and then it will be crosswalked over into the main database as soon as possible. We've been getting more efficient and faster at this over the years, so it doesn't really take that long. But if you are in a time crunch, you slept in, and you need to have your data in from your partners by the end of the day or the end of the week or something, you can always reach out to us and let us know that you need data to be crosswalked over as soon as possible, and we can help expedite that process. Like I mentioned before, you want to mark everything ready for upload. You want to go through and review all of the data beforehand. So you can start out by going into the AGOL group for IMAP Mobile Advanced, and there is a help document there, IMAP Mobile Advanced Tips for Data Collection. This has a little bit of advice on pre-survey, during surveys, and then post-survey data review as well. So going through that initially, and then just a few extra tips here for general data QA/QC. For the most part, this applies to the IMAP Mobile Advanced data, but a lot of this can also be applied to data that you have collected and put in with other tools. You're still able to edit your data once it has been put into IMAP before it is confirmed. We'll go over the confirmation process a little bit. But when you first upload your data into IMAP, it is unconfirmed, and you're still able to edit it. You can change, species information, comments, or location, or shape of polygons, things like that. So starting out with searched areas, making sure that the searched area has been shrank down to represent the actual area that you surveyed. Oftentimes we recommend people start out with large searched areas to encompass the entire area that they might be working in, and then shrinking it down later to more accurately represent the area that you're actually working in during the season. This is especially important if you're going to be using like the acreage from these searched areas in your end of your metrics for how many total acres you surveyed or did management on. Filling out all relevant fields. So the searched areas have a lot of optional data fields that you can fill in for things like crew hours or crew members that are working on the site. So if you have metrics that you want to include in these searched areas, you can do so. And then double checking all of your numbers associated with them. So that's your organization ID number and your personal person ID number for your IMAP account. And this is to make sure once everything gets crosswalked over into the IMAP invasives database proper, everything will be associated to the right organizations and the right people for observations. For the different features that you were collecting in those searched areas, your points, lines, and polygons for presences, treatments, or detected records, making sure that every feature that you put down on your map has a species associated with it. So, you get your searched area, you go to the linked polygon, and then going into that polygon and making sure that there is at least one species linked, one or more species for each one of those. If you have any test records or placeholder records, going through and deleting all of those. So an example over on the right here, where I made a little test searched area and polygon point and named it test delete, making sure you go through and delete all of those and don't mark them as ready for upload. If you don't want them in there, just getting rid of them is the best way to do that. Also, if you were out in the field and you found any, place of interest that you wanted to make a point for to go and revisit later or to save the GPS coordinates for, going through and making sure you have that data and putting that into a secondary location. Or any like fake species record or anything like that, we do go through periodically, usually about once a year, and we delete all of those off of the IMAP database. So if you have any important information for like a rare plant or a spot of interest in one of those fake species records, making sure that you get that data and put it in a secondary location. Next, making sure that all of your features are correctly linked to one another. So if you have, you went in and you did management for Japanese knotweed or something like that, making sure that your treatment polygons have their Japanese knotweed species associated with them, that there is a presence polygon for Japanese knotweed again, underneath that treatment, and that all of that is correctly linked to the searched area for the and then any complex geometries that you may have, going back in and fixing any of those. We understand working out in the field might not be ideal conditions, and sometimes your polygons can come out looking wonky, and you'll say, fix that in the office on a later date. So before you go in and submit all the data, making sure that you go through and fix any of those wonky polygons. So a few examples here. If you have a treatment polygon that doesn't really quite match up with the presence that you initially had, kind of buzzing those around or editing the shape to make sure they line up a little bit better. Ensuring that all of the data you've collected, not just polygons, but lines and points as well, fall entirely within the searched area that you were working in. And then if you had any polygons that crossed over each other, so for this example, this is one singular polygon, where as it was being drawn out, it kind of crossed over itself in vertices and ended up with making two small separated polygons that are actually one feature. So if you have any cases like this, going back in and making sure that you can, pull those vertices apart and make it actually look like polygon. Features like that don't really play nice with the crosswalking, so we'll have problems getting those in. Similarly, if you have a streaming function for any polygons that you were taking, where you can just stream your location and walk around like a population of whatever species you're trying to mark down. Sometimes if you stay still for too long, it can make a bunch of vertices in one location and kind of cross over each other. Having instances of that, going in and making sure you clean it up as much as possible. If any of these problems do make it through your review and we catch them on our end, we will reach out to you and let you know about them. And to talk a little bit more about that, I'm going to hand it over to Fate, who's going to talk about the IMA review process, which has had some changes recently. Fate?
Yeah. Thanks, Doug. I'll also point out, Doug, you're at like 95% audio strength, so you might want to tweak a little here while I'm chatting.
Sure.
Yeah, so I just wanted to mention to everybody, particularly for those of you who are using IMA, that we have made some changes to our sort of IMA review workflow. In the past, it's kind of gone from a fairly manual process to, in the last year or so, sort of a semi-automated process. But now, our colleague Zach Davis has put together a script that almost fully sort of streamlines all of the various checks that Doug was talking about before. So it really speeds things up for us, which is really nice. And so, essentially the script is going to flag these errors. You can see on the right here, our review glossary. And basically, if any of your searched areas or polygons or whatever, presences, treatments, whatever, have issues, the script will flag those errors, and then basically I will sort of reach out to you, to various organizations with the records and sort of the errors they have. And then we're also including in this glossary sort of our recommendations for how to handle those issues. I'll add that hypothetically, this might mean that you may be seeing a few more sort of error records pop back in your inbox from me, because historically I've been doing a lot of manual nudging, fixing of attribute data here and there or polygon stuff. I will be doing less of that moving forward. So you may get a few more errors than before. So just for us, emphasizing that this data review process is really helpful and can sort of make everything go faster. So if you don't want a bunch of emails from me, definitely, check on all these things before saying ready for upload. And then importantly, lastly, this is a new part of the process. If I reach out to you with any sort of revisions you need to make, once you have made those revisions, you can just mark the data as ready for upload again, and that will, signal to our system to run the review again. So yeah, in the past, maybe you've kind of emailed back and forth with me, but you can still do that. But mechanically, the most important thing to do is to mark your data as ready for upload again once you've completed any revisions. All right, that's all for me.
Perfect. Thanks, Fate. Yeah, and if anybody has any questions about the IMA data review process or any of that, you can put them in the chat now again, or you can reach out to Fate at a later date. All right. So with that, we're going to move on to the data confirmation process. So now all of your data that you collected over the field season has been successfully moved from your mobile devices into the database proper. As most people know, when you first are submitting data to IMAP invasives, in all cases, it comes into the database as unconfirmed. Unconfirmed meaning that it's not publicly viewable. If you just launch an instance of IMAP, you won't be able to see that data right away, but you will be able to see your own unconfirmed data and other people within if you're part of a organization or a project that you were collecting data for, those other members will be able to see the data as well. But that data remains unconfirmed until it gets reviewed by an expert, again, with the exception of just the IMAP Mobile Advanced data. As that is crosswalked into IMAP invasives, that is confirmed automatically via partner data. And I will make a note about that in a little bit. This is just what it looks like in the main database view. So if you're looking at just your data, if it's popping up as green, that means it is confirmed. It is viewable to the public. Anyone with or without an account can see it. And if it's pink like this, that means it's unconfirmed and only you or other people in your organization or projects are able to see it until it has been confirmed by an expert. So when I say expert confirmation, what do I mean by that? So different people in the invasive species community, in the professional agencies and whatnot, have certain permissions to confirm the data for certain species. There are select few species. For the most part, it is high priority species. For the most part, that's insects or high priority plants like giant hogweed, things like that, that we have specific agency staff members that go through and do the confirmation process for it. So an example of that would be like spotted lanternfly. All of those records get confirmed by members of the Department of Agriculture and Markets. For all of the other various species, this is confirmed by various agency staff, people on, the DEC or state parks, various professionals who have permissions to confirm the data for species that they are dealing with or experts in. And then organization members can also confirm data. So if you have an organization or a member of an organization, like a soil and water conservation district, for example, you can request permissions to be able to confirm data for just your organization. Anybody can request data confirming privileges if you are, again, if you're an expert in the field, if you work in invasive species, or you think that your skill set is good for confirmation of data, you're really good with your species ID. You can reach out to us and confirm or request confirming privileges. And again, this is done on either an organizational basis, where you can confirm data that is just tagged to your organization, or on a statewide basis. This is usually reserved for people who are specifically in the professional community. And this will allow you to confirm data for certain types of species anywhere in the state. So the way to get these confirming privileges and to see a review of the entire data review and confirmation process is via our data review help document in our help resources library. The link is here, and I'll also include a link at the end. But if you go into this document here, it gives you all the information about how the data review process works, and it will give you a link for the data confirmation request form, where you can basically just put in your information, who you work with, what are your qualifications for confirming data, and which of the tax type you would like to have data confirmation privileges for. And that process is the same for either your organization or on a jurisdictional level. So if you want any sort of those permissions, go into that document and fill out that form. And that is it for data confirmation. And we'll jump into data analyses. So all your data is off your tools into the IMAP invasive database, and now it's all been confirmed and verified, quality controlled. All the information you have in there is now 100% perfect, good invasive species data. The next question is, well, what do we do with that data? This is a question that we at the database get all the time. People will call us and say, well, why am I bothering to report spotted lanternfly if you're not going to come to my house and kill them all? And the answer that we give is all the data that is contributed to the IMAP invasive database helps with statewide analyses. And these analyses are important for things like prioritization and helping local and regional land managers to know where they should be prioritizing their resources, their time, which areas are most important, and aspects like that. One of the main ways we do this is through our tiers analysis. So we have our various invasive species tiers, tiered 1 through 5. And basically what this is we take all of the information from the entire invasive species database and we run it through an analyses process where we are looking at the abundance of data we have for each species, their general invasiveness ranking based on the ecological and socioeconomic impacts they have. Um, and we're basically saying how much of a priority we believe based on the data that each of these species is. So this is something that we run statewide annually, and we also break it down into a regional level for the 8 prisms throughout the state, and we give them the data for them to look at and make their own assessments if they want to make any changes to the tiers year after year. If anybody was on the Prism call earlier today, you may have heard several of the prisms mention that they have completed their 2026 tiers analyses. So the numbers for their tiers for their species in their region will be changing. If you want more information about the tiers, we have a whole webpage devoted to it that has the entire tiers table and also has links to things like story map that go into more detail on how the tiers analyses process works. Well, this is just a quick look at the tiers table. I've got a few things checked off here. I've got the statewide tiers, the Finger Lake Prism and Western New York Prism tiers for all species that are tiers 1 or 2. And you can look here and see some of the differences in it. If we take an example like Brazilian waterweed here, you can see that it's a high ecological impact. It has a negative impact, but not a significant one. Across the entire state, it's considered a tier 3 species, so it's in several locations, and eradication isn't really a feasible strategy anymore, but containment is the main strategy. In the Finger Lakes region, it's a tier 1, so it isn't found anywhere in that region, but they're on the lookout for it. And in the Western New York region, it's a tier 2, which means it's found in a couple of locations, but is still small enough in abundance that eradication and containment is still a feasible goal for it. So all the data that comes in gets run through this process on an annual basis to help prioritize, where the species are, how abundant they are, and which ones should be prioritized for management. Another analysis we do is more specific, and that goes through our various analyses and prioritization tools. So similar to the tiers process, we update these data sets on an annual basis, usually throughout the winter, so they're ready for the next field season. So all of the data that you're collecting throughout the summer of 2025 should be ready in these various tools sometime in mid-2026, usually right before or at the beginning of the field season. So we have a number of data analyses tools that are good for different specific areas. Things like our prioritization mappers for aquatic invasives and terrestrials. Here in the bottom right here, this colorful map of New York State is an example of the terrestrial invasive species prioritization tool. And just to go through quickly how that works, there's 3 different data layers that get aggregated into a main data layer based on a whole bunch of different factors. This is using data from IMAP Invasives, New York Natural Heritage Program, and a bunch of other different data sources. So just to break it down, red layer is risk of spread, and if you take a look at the map there, you can see that the darkest red areas that are around things like urban centers or major roadways that introduce risk of spread. Blue areas for protected areas, so this is things like state forest preserves or state parkland, basically anything that is not privately owned or developed land. Green areas for ecological significance, so this is yielding or heavily from the New York Natural Heritage Program data layers. So you see like a dark area up in northern New York there in the Chameau Barrens area or the Alvar type ecosystems, which are ecologically significant and sensitive ecosystem types. So those are considered to be of high ecological significance. And then the final layer is a total score. So that's all of those other data layers layered on top of each other to see which areas are overall most important for prioritization of invasive species surveys or management. And if you take a look at here, you'll see the areas that are in dark orange or red, places like the coastal wetlands or marshlands of Long Island, High Peaks region, or the Catskill Preserve. So these areas are, they kind of hit the trifecta of high risk of spread, protected areas that are available for preservation, and high ecological significance. Some of the other analyses tools that we have here back on the left side, we have the Rare Invasive Species Mapper. Like I mentioned earlier, New York Natural Heritage Program is data collectors for all of the rare, threatened, and endangered species. So we've taken that data set and we lay it on top of the invasives data set and kind of see where those intersections are. These are considered to be very high priority areas, so we want to make the proper parties aware of them. There's an asterisk next to it because this is obviously it's very sensitive data for all of the rare species. So this is given out to a select few managers throughout the state to base their prioritization on. We have our water body lookup tool, which is up in the top here. This takes from the database all of the aquatic species. So if you want to know, like, for the Hudson River in Westchester County, a full list of all the species, you can do that. Or if you want to know all of the water bodies that have banded mystery snails in it, you can just choose that and it will give you all of the water bodies and all the counties where that's found. Basically gives you a really quick synopsis of specific locations or specific species. We have our management outcomes viewer. If you've been going back to the same sites year after year and collecting presence and management data and conducting treatments, and you've been putting all that information into IMAP invasives year after year, we've been running that through this analyses tool, which will give an outlook on if these sites are trending towards eradication, if the relative abundance or size of populations has been growing or shrinking over time, and generally if it is getting better or getting worse for these sites. So if you're somebody who manages a lot of different sites throughout your region or throughout the state, this is a good tool for that. And a slightly more specific version of that is the water chestnut action sites, which just works for water chestnuts. So similar to the management outcome viewer, it takes all the data that is input year after year for management of water chestnut. It takes the size of polygons, the relative abundance of what was recorded during management efforts, and it can give things like biomass estimations for estimates of how much water chestnut is in the water body. There's an asterisk next to that one because that is a dashboard that is available on ArcGIS Online. So you need to have an AGOL account and we can add you to that group. So if you're somebody who works for a professional agency where you have an AGOL account and you work specifically with water chestnut, this might be a good one to reach out for us to get you added to that to look at your various sites throughout the state. So those are the data analyses that we offer on our end, but if you want to conduct your own analyses, we have multiple tools that allow you to do that. First off, the main one we have is the export and report function. So starting out with exporting, if you want to take the data right off of IMAP invasives and put it into your own, or excuse me, starting with reports, not exports. If you want to, run reports for the data that we have on our website in the database, we have options for running reports for 4 options by default. You can do a species list by geography, so all of the species that are found within a certain property or county or area of interest. We have approaching region species, so if you want to know what isn't found in your PRISM region but is in neighboring PRISM regions, you can run reports for that. Infested areas, which runs total acreage analyses of certain species within a certain area, or area treated, which is similar to invested area, but for treatment data. So you're allowed to go through and do all of the different fields, set your area of interest, what kind of reports you want to run, what kind of data you want, if it's for present species, unconfirmed treatments, whatever you want to do for that. iNaturalist data, and I guess a quick point about that. If you or any of your volunteers are contributing data to iNaturalist for invasive species, that can get crosswalked automatically into the IMAP invasives database, so long as the records are research grade. So 2/3 of people agree with your taxonomic identification that you've said for what species it is, and if you have your data sharing enabled. So there is a layer in the IMAP invasives database for all of the iNaturalist data that hits those criteria. And you are able to include that data in these reports as well. And then over on the right side here, we have an example of the infested area report. So I just kind of drew an ambiguous searched area in yellow here around a few polygons for various species, and it will give you a breakdown of how many species were within that area, how many acres of polygon data there is. You can also set it to have collection of any points that are in that area, and it will assign a area value to it. So you can see up at the top there the point buffer size of 5 meters. So each of the points will be buffered with a 5-meter diameter around it, and it will give you total acreage for all of the various species within that area. So this is good if you need to pull metrics quick from the data and you don't want to export it all out and make your own maps with it. You can run one of these reports to get quick numbers for, abundance of certain species in your area or total acreage. You can also, like I mentioned, export the data out of IMAP invasives to put into your own programs or conduct your own analyses on. So the 3 ways you can do that is through CSV files, which comes up as Excel files, shape files if you want to put them into your own ArcGIS layers, or if you're like me and you don't like change and you're still using the GIS ArcMap desktop version, you can put it into that, or a geodatabase if you want to add it to your own database layers. Very similar to reports, has a very similar looking window. You choose what export type you want. Filtering data, so if you only want specific species or specific taxa type, you can filter that out and enable the filter. Setting your boundary, again, for your property of interest, or here we have the entirety of Herkimer County selected, and then exporting whatever data you have selected there, and getting those files that you can download onto your machine and do whatever you want with them. So over on the right side here, I have an example of a data set for treatment records at Caroga Lake State Park, and then the various different species where I went into GIS and made them different colors. This is good for if you want to provide maps for any year-end reporting, or if you're working with partner agencies and you want to provide maps for them. This can also be helpful for visualizing general abundance of where things are located, how abundant they are at first glance, if you want to prioritize which areas you want to manage in the future. If you want to get a basic map and you don't want to spend all the time exporting the data and putting it into GIS and color coding it and everything, we have a built-in ability to do similar functions in the distribution layers. So in the main IMAP invasives map viewer, over in the layers on the right side, there is a distribution layer tab that you can open up. And what you can do here is you can set your distribution. So you can do this by county, like I have here on the example. You can also do municipalities, water bodies, hydro basins, prism regions, whatever you want to do. And it will give you a basic yes, no, if certain species are found in that area. So for this example, I have just mile a minute in the state of New York filtered on, and then I hit the show distribution layer, and all of the counties where there is mile a minute data in our database, they all will light up in orange. And this is good for just like taking a quick screenshot and including it in a report if you just want a general overview of how abundant some of these species are. You can also remove them, you can give them unique names, and you can save them to your distribution layers. So if you want to run multiple, kind of layer them on and off, you can do so. So just a quick example here, I have my mile a minute layer that I had in the last screenshot, and then I also ran a hydro basins distribution layer for hydrilla, and I can layer that on top. So I can see, where those two intersect if you wanted to do that, or If you were looking at where they cross over state boundaries or which counties are impacted, you can do that for any species, any combination of species, and any geographic distributions that you want. And then lastly, I want to talk a little bit about the time series. So this is a button that's on the top bar. Create records, filter records, identify, and then set time series. This is a button that you've probably seen a million times, but not really used all that much. And this is good for showing a time lapse of the data at a specific site. This works if you are zoomed in pretty close and you're at least far enough where you can see the specific data points instead of those larger yellow aggregates of data. So if you're able to see the green data points, lines, and polygons, you're able to use the time series tool. And to show you how this works, I have a time series set up for Slender Falls Brome Records, and this is the lower half of Letchworth State Park. And I have a series set up between the years of 2018 and current day. And if you run the time series, this is just what it looks like. This is a screen recording. It will scroll through each year as the data comes in, and it gives you a little video of what it looks like as the data rolls in year after year. And you can play around with this. This works for presence data, not detected records, as well as treatments. You can also set it for just showing data that has been contributed within a year. So all the data within 2018, then just the 2019 data. So if you're going to the same site year after year and you're resurveying the whole thing, or you're conducting management year after year, this could be good to show the change in number of acres that have been shown to have a certain species or things like that. All right, and then again, a few times I mentioned that there are a few help documents or resources that help walk through some of these steps. All of these available on our website. You can go to the nyimappingbasics.org training page, or most of the pages on our website have a link to this directly in the upper right corner. On that resource library, you can search for whatever documents you are interested in. We have documents for all the specific data collection tools, like I mentioned before, the confirmation process, running reports, as well as various other aspects of using the IMAP Invasives database. If you're looking for something specific and you aren't able to find it, you can always reach out to us directly, send us an email if you're looking for specific